Howdy, folks. Thanks for checking into Mr. Ulrich's landofbiology.com. I am Mr. Ulrich. In this notescast, we're going to be talking about how we go from one long strand of like lots of base pairs to two long strands of lots of base pairs. Sometimes when people are asked to think about the structure of DNA, the names that come up are James Watson and Francis Crick. And they certainly did write the article in '53 that described the double helix structure of DNA. But of course, if it weren't for the work of Rosalind Franklin, they wouldn't have had the information or the input necessary to make that model in the first place. So really, it was the Franklin with Watson and Crick model of DNA. Regardless of whose name should be on the model, when Watson and Crick published their '53 article on the structure of DNA, one of the first things they noted was the way that it kind of lended itself, the structure of the molecule with this double helix and the way the bases were held together, the base pairs, I should say, were held together, that it lended itself to an elegant mechanism for unzipping and copying and holding a sequence, a code in their structure. I'm hoping that as AP students, the idea of DNA being a molecule for encoding genetic information in a sequence of bases is not new to you. When you learned about that, your teacher, maybe me, probably related that encoding of information in a sequence of bases to encoding information in a sequence of letters as a sentence. And just like you need to read a sentence from beginning to end, you need to read DNA from beginning to end. So there's a way to kind of start a sentence with a capital letter, and there's a directionality as well to DNA. Now, the way we look at directionality in DNA has to do with the way we refer to directionality, I should say, has to do with the number of carbons on the carbon sugar. And the first carbon sugar is the one that is in the carboxyl group in the aldose form. Not that you'll need to know that. But you start with carbon number 1, and then you just number them 2, 3, 4, 5. This means that the side chain of a strand of DNA, or the backbone of a DNA strand, is alternating phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. And when we want to talk about the ends of DNA, of a DNA strand, we talk about the 3' end or the 5' end. The 5' end is the one that has the phosphate attached to it. You can think of it as this whole structure here being one nucleotide, and then each other one is attached to that one. So the 5' end always has a phosphate group attached to it. It's the phosphate of that particular nucleotide. And then the 3' end won't have anything attached to it. It will have a hydroxyl group attached to it, so you can do some dehydration synthesis and add another nucleotide on the end if you are extending the chain. But that is the 3' end of that piece of DNA. We always are adding a phosphate onto the 3' sugar of the next nucleotide. DNA exists in a double helix, so it's not going to be in one strand. It's going to be 2 strands kind of attached to one another, and they run, though they have directionality, they run in opposite directions to one another. So we call that anti-parallel when the complement runs in the opposite direction. So one side will run from 5' to 3'. The other one will kind of be on its head upside down. It'll be running from 3' to 5'. It's important for us to recognize the different bonds in DNA and how they lend themselves to the function. There are stronger bonds and weaker bonds. The bonds holding the bases together are hydrogen bonds, and as we've talked about, those are pretty easy to pull apart. But the bonds holding the side chains together and thereby holding the sequence of DNA together are much stronger covalent bonds. We call them phosphodiester by the way that it's carbon-oxygen phosphate. Phosphate, oxygen, carbon, right? That kind of looks like 2 ester bonds. They're not quite esters because of the phosphorus, so that's why they're phosphodiester bonds. But those being strong, conserving the sequence of bases, where the hydrogen bonds are weak, allowing the 2 strands to come apart. Again, I hope the idea of complementary base pairs is not new to you. So this whole idea of A's and T's and G's and C's, right? Not a new thing. There are two different types of bases. We have of nitrogenous bases. We have the purines, which are adenine and guanine, and we have the pyrimidines, which are thymine and cytosine. Now, we always get A's and T's because we always get a purine with a pyrimidine. Turns out there's only room for 3 little rings in between the side chains, and so you're always going to get a 2-ringed purine with a 1-ringed pyrimidine, so you have those 3. Now, the reason why we always get A's with T's and not A's with G's, right, has to do with the number of hydrogen bonds that are formed between them. A's and T's form 2, right, where C's and G's form 3. This, of course, means that if there are happen to be more CG pairs in a molecule of DNA, it's going to be more stable. It's going to be a little harder to unzip than one that has more ATs. If we're trying to divide cells, then we're going to need to make a copy of DNA. And this is called replication because when DNA copies, it unzips, and then you end up making 2 strands from the unzipped DNA. So one strand serves as a template in both new strands. So I don't end up with a copy and an original. Each strand is part copy and part original. We call that semi-conservative. Now, DNA replication doesn't start at one end and go through the whole molecule. It actually kind of starts in the middle like a zipper that's broken and unzips in the middle, right? You're going to have several of those along a strand of DNA so that it doesn't take as much time for this to happen. We call those replication bubbles. Now, the first step that's going to take place, we have to unwind the DNA because the DNA all coiled up. It's actually wound up around nucleosomes and, well, all kinds of stuff. But anyway, we have to unwind that DNA. And this isn't going to happen on its own. It needs a little help from an enzyme. In this case, the enzyme is called helicase. Helpful because it deals with the helix, right? Now, as that helicase works, it's actually, there's going to be one on one end and one on the other end, too, of this bubble because it's going to grow in both directions. In order to keep the keep the bases from coming back together again, right behind the enzyme, there are these proteins that are kind of along the way there that stabilize each strand apart from each other. Now that the DNA bubble is formed and I have all those exposed nitrogenous bases, to build the daughter strand, it's just a matter of allowing the complementary nitrogenous base-bearing nucleotides to bond into position. This is going to happen on its own. It's going to need an enzyme. So along comes DNA polymerase 3. Now, it's not important at AP level for you to have all these enzymes memorized or anything like that, but it's always good to be familiar with what is actually going on. So DNA polymerase 3 is going to allow the complementary nucleotide with the proper base to bond into position, get 2 of them into position, and then bond those guys together, and then scoot on along, adding the next one and adding the next one. Now, taking these molecules and organizing them together, bonding things together, synthesizing new DNA, right, is endergonic. It's going to take energy. And where is that energy going to come from in order to allow this process to take place? And I don't know if you've noticed, but adenosine or adenine should sound familiar, right, since we've already talked about ATP. ATP is it's a nucleoside because it has a nitrogenous base, in this case adenine. It's got ribose sugar and it's got a phosphate group, right? There's your nucleotide, and then it's got 2 more phosphates added to it, right? All right, so the ATP kind of comes with its own energy, and just like I can make ATP with adenine, I can make GTP with guanine, and TTP with thymine, and CTP with cytosine. And so each one of these nucleosides is what's actually added. We turn it into a nucleotide once it's there. So the DNA base kind of arrives with its own energy power kind of intact. And that energy power is used by DNA polymerase 3 in order to add them together. So replication is all about adding the right bases. Now, the trick with a DNA polymerase, whether it's 1 or 3, is that it can only add bases because it's adding one with 3 phosphates attached to it. It can only add them to the 3' end, because that's the end that doesn't have any phosphate. It's the end that's going to get a phosphate, right? So the problem, of course, is that you need, and we're going to talk about this in a moment, you need some sort of starter, because DNA polymerase 3 needs a 3' sugar there in order to bond to. Right. This way, the strand, I should say, only grows from the 5' end to the 3' end. So it's kind of being added onto that 3' end. And the DNA polymerase 3 comes in and it's going to clip those two phosphate groups off. There's the energy, and use that to bond the phosphate to the sugar. And then with the next base, it does it, and it moves down a little bit more to the next base, and it moves down a little bit more to the next base. Each time the base comes essentially with its own energy source. When we look at the bubble itself and the way that the bases are added, now that we have directionality and we have this problem of only being able to add onto the 3' end of the strand, it's going to cause some issues. So here's our helicase with the stabilizing proteins in place. Right, and here's the references for us with directionality, as directionality goes, giving you the 5' and the 3' end of each stage, each strand. So DNA polymerase 3 comes along, and it's adding on here from the 5' end towards the 3' end, so it's adding on to the 3' end, then that's okay, that works. But up above, it's got to be able to do the opposite. Up above, it's got to go kind of, well, now it's got to add onto the 5' end, and we know that that doesn't work. So the way that it gets around this is by just doing it backwards. It binds a kind of small group and kind of bonds that into the 3' end. Right, so this way it's still adding on to the 3' end, and it just bumps into the next one. We call these little short strands of DNA that are added. They're called Okazaki fragments. Named after Okazaki, who ran the team that made the, who discovered it. Now I have these little fragments, I got to add them together. And of course, adding them together involves messing with that 5' end of the other strand. Now, because one side goes really fast, and the other one is done with these little strands that are going to get bonded together using this enzyme ligase. One strand is called the leading strand because it has continuous synthesis that's going on, and the other one's called the lagging strand because I got to build the Okazaki fragments one by one and then bind them together with DNA ligase as the spot welder. Now we look at the replication bubble itself. All right, as it unzips, we actually get a fork kind of at either side. And because, again, it's anti-parallel, what's going to be the leading strand on one side is actually going to be the lagging strand on one side for one half of the bubble. When you look at the other half of the bubble, what was the leading and lagging strand on the one side becomes the leading and lagging strand on the opposite side. The problem we haven't addressed is how does the leading strand get started if it needs a nucleotide with a 3' end already in position? Well, that's going to be done by the use of RNA. So instead of building a little DNA, we're going to use an enzyme to build a little RNA primer. It's got a complementary base sequence, and it sticks there for a little bit, right? And that's going to give something for DNA polymerase to add onto to start building that. That, of course, means that each one of the Okazaki fragments starts with a little RNA primer piece. And when it comes time to, bind those together, we're going to run into a little bit of problem. It's the enzyme primase that builds that starter sequence for the DNA polymerase 3. Now, when it comes time to actually go in there and allow ligase to do its job, it's got to first kind of remove those RNA nucleotides and replace them with DNA nucleotides. That's going to be the job of DNA polymerase 1. All right. Again, it can only go in one direction. So it's going to be able to pull these nucleotides out pretty well and allow DNA ligase to do its job. Then it's going to go down to the leading strand and chew off the end of that. But again, DNA polymerase 3 can still only add bases onto the 3' end. And down on that leading strand, the part that is exposed is actually the 5' strand. And so you're going to end up with some chromosomal erosion taking place every time there's a cell division, right? Because each time you lose a little bit of that leading strand, and you lose a little bit of the DNA code. All right. Now, does that give us a limit to the number of divisions a cell can actually go through before you start losing information, actual information from the chromosome itself? Well, there's an adaptation that has developed that allows for a little bit of a buffer zone for this chromosomal erosion. There's a sequence of it's like pawns, right? They're designed to get chewed up. They're just non-coding sequences of DNA that act as a protective cap. But even with this in position, some cells are still limited to about 50 cell divisions. And you get these little repeating ends. And where do they come from? They're actually built by an enzyme called telomerase. And telomerase will add DNA bases onto the 5' end of the DNA and replace the parts that are actually lost. Now, what types of cells might have higher activity of telomerase that's going to kind of rebuild those telomeres? Right, well, stem cells certainly have to do an awful lot of dividing, and so they're going to have an upregulation in those telomerase genes. And cancer cells, unfortunately, also divide and divide and divide and divide and divide, and will often have upregulation of telomerase genes. Just to quickly review that replication fork, right, there is a directionality to them. There's going to be one on the other side of the bubble, but when we look at the single direction, helicase unwinds where those single-stranded binding proteins are going to kind of stabilize it so it doesn't just stick back together again before primase has a chance to add the RNA primer for those Okazaki fragments, and DNA polymerase gets its chance to add the DNA nucleotides to either the leading strand or those Okazaki fragments themselves. And ligase is going to come along once DNA polymerase 1 replaces the RNA primers. Ligase binds them together. Of course, we don't see that in the leading strand because it's just getting added onto that 3' end. But in the lagging strand, we see all this activity. That's why it's called the lagging strand, because it has all of this action going on around it. Now, with all this action, with all of these bases, and because this is being done by stupid molecules that aren't capable of reading and proofreading, it would seem, there is a lot of proofreading that actually goes on because you would expect a lot of typos, and there are. All right, now DNA polymerase, because it can go through and replace nucleotides, it can see where there's a bulge, either through a thymine dimer where two neighboring thymines bond to each other rather than to the adenines across from them, or a place where you do get two purines together and you get a bulge going there because there's 4 rings in that spot. DNA polymerase 1 can come by and clip out the offending base and replace it with a nucleotide with the appropriate base. It's pretty amazing at the amount of error reduction that these, again, stupid molecules are capable of achieving. It's quick. It has to be quick, even for the small genome of E. coli at 5 million base pairs. They do their division in a pretty short period of time. All right. When we look at human cells, they divide very quickly as well. Once every 20 minutes or so is not unusual during the first stages. And that's a lot of bases because we have a much bigger genome than E. coli goes through. But even so, it is remarkably accurate with only about 1 error per 100 million bases. Though those do accumulate over time, it's still, especially given all the things that can go wrong, it's amazing that we do get up in the morning. To take a look at what a replication bubble actually looks like, here's 4 different replication bubbles. It's a little hard to see. That's why there's a little guide there over to the right. But you can see that there's numerous places where helicase is doing its job and all the other aces are working away. We'll leave it there. Again, thanks for checking in to my videos. I do appreciate it. And if you have any questions, feel free to leave them in the comment section if you're on YouTube, or send me an email, or if you're watching this because you signed it, sorry, write me a question, and we'll see you in class.